Continuar con la siguiente charla, que corresponde a la doctora Zakreski. Voy a leer su reseña, por favor. La doctora Zakreski es investigadora de ciencia básica en neurociencia y cognición en el Departamento de Psicología y Ciencias de la Vida de la Universidad Charles en Praga, en República Checa, así como investigadora en el Centro de Salud Sexual e Intervenciones en el Instituto Nacional Checo de Salud Mental. Sus intereses de investigación se centran en las respuestas neuroendocrinas al estrés, la adversidad en la primera infancia, la estrategia de la historia de vida y la formación de vínculos en la infancia y la adultez. En particular, estudia cómo las diferencias individuales en la reactividad al estrés median la depresión o la resiliencia y alteran los procesos de apego. Su investigación actual examina factores etiológicos en hombres con intereses sexuales violentos y parafilias, y el papel de la sincronía conductual y emocional en la salud del apego sexual y romántica. Es originaria de Calgary, Alberta, en Canadá. Obtuvo su doctorado en neurociencia en 2020 por la Universidad McGill en Montreal, Quebec, Canadá. Le damos un aplauso a la doctora.
Thank you for having me. It's my pleasure to talk to you today about the crosstalk between the hypothalamic pituitary adrenal axis and the hypothalamic pituitary gonadal axis, and the relevance of this crosstalk to understanding altered sexual behavior in individuals exposed to early life adversity. Can you hear me okay? Good. Pointer.
You can just...
So I point this way. Okay, can you hear me now? And the pointer towards the computer. All right. Closer? How's this? Cool. So I'll be talking about how early life adversity affects sexual behavior, some of the physiological mechanisms underlying this association, and why early life adversity affects sexual behavior. So what is early life adversity? It comes in many forms, such as physical, emotional, or sexual abuse, neglect, exposure to violence or conflict, parental separation or loss, living in an unstable, unpredictable, or chaotic environment. Basically, any experience or event that leads to very high or chronic activation of the physiological stress response systems during child development. The physiological stress response systems change physiology as to enable the organism to survive challenge and to avoid disruptions of homeostasis. So, for instance, coordination of the fight or flight response. There are two key physiological stress response systems. So you have the autonomic nervous system, which mediates faster but shorter lasting effects. The hypothalamic pituitary adrenal, or HPA axis, is slower and has longer lasting effects. Now, while both systems are important for stress-related illness, historically, the hypothalamic pituitary adrenal axis seems to be particularly important. The HPA axis is a highly conserved neuroendocrine circuit involving the hypothalamus, pituitary, and adrenal glands. So stress activates neurons in the paraventricular nucleus of the hypothalamus to release corticotropin-releasing hormone, CRH. CRH then travels to the anterior pituitary, where it stimulates the release of ACTH. ACTH then enters the blood supply, travels to the adrenal glands,
and stimulates the release of glucocorticoid, steroid hormones. So in humans and some species, cortisol is the primary glucocorticoid, whereas in other species, such as rodents, corticosterone is the primary glucocorticoid. Glucocorticoids then travel throughout the body and provide negative feedback onto the HPA axis, thus decreasing further HPA axis activation. CRH, ACTH, and glucocorticoids affect the brain, changing learning, attention, and emotion in ways that are generally adaptive when facing acute stress. Glucocorticoids affect numerous systems throughout the body. Most tissues in the body express glucocorticoid receptors. During stress, glucocorticoids suppress inflammation. They make energy more available for immediate use. They also inhibit non-critical functions, such as growth. The HPA axis also has important effects on reproduction. In humans and animals, activation of the HPA axis generally inhibits reproduction. So hypercortisolism, due to Cushing syndrome or chronic stress, can lead to sexual dysfunction, amenorrhea, or prevent ovulation. Evolutionarily, it makes sense to inhibit reproduction during stress because stressful conditions are not a good time to reproduce. Inhibiting reproduction also could free up resources to be used for immediate survival. But we'll talk more about whether this makes evolutionary sense for everyone. So the effects of stress on sexual function and reproduction largely involve the HPG axis, the hypothalamic pituitary gonadal axis. This neuroendocrine system is critical for regulating sexual development and function. There is extensive crosstalk between the HPA axis and the HPG axis. Understanding this crosstalk is critical if we want to understand the relationship between stress and reproductive function. The HPG axis, like the HPA axis, is highly conserved across vertebrates. It's a neuroendocrine circuit involving the hypothalamus, pituitary, and gonads. So neurons in the hypothalamus release GnRH, as we already saw earlier. GnRH then travels to the anterior pituitary, where it stimulates the release of gonadotrophins. So you have luteinizing hormone and follicle-stimulating hormone. These gonadotrophins then act on the gonads. Luteinizing hormone stimulates the release of the gonadal sex steroids, such as testosterone, estradiol, and progesterone. Testosterone gives negative feedback, whereas estradiol can have either positive or negative feedback on the HPG axis. We'll go to the next slide eventually. Okay. As I mentioned earlier, the HPG axis is critical for regulating sexual development, reproduction, and behavior. The HPG axis promotes fertility, stimulating spermatogenesis and ovulation. It's also critical for the development of primary and secondary sexual characteristics. It is involved in sexual differentiation of the brain, which has important implications for sexually dimorphic behaviors. The HPG axis is also important for the organization and activation of stereotypical sexual behavior in many species. So for instance, lordosis and mounting in rats. The HPG axis also affects behavior and anatomy in ways that would facilitate competition for mates. So for instance, testosterone is associated with aggression. The HPG axis is also critical for sexual motivation and desire. So we know that sex steroids increase attention and responsivity to sexual stimuli. The HPG axis is regulated by complex afferent networks, which integrate external and internal cues so that the HPG axis activity is optimal for the current conditions. So for example, we integrate information about energy balance, photoperiod, pheromones, and stress, which I'll talk to you more later. So there are several key regulators. So kisspeptin neurons generally have an excitatory effect on GnRH neurons, and they're also very important for stimulating and maintaining this pulsatile release of GnRH, which is necessary for normal reproductive function. Gonadotrophin inhibiting hormone neurons, or in mammals, RFRP neurons, obviously have an inhibitory effect on the HPG axis. You also have GABAergic neurons that are projecting to GnRH neurons. These can have either an inhibitory or excitatory effect on the HPG axis. So each of these regulators are actually important for understanding the effects of stress on the reproductive axis. The HPA axis, both acutely and chronically, generally has an inhibitory effect on the HPG axis. This occurs on multiple levels. At the level of the hypothalamus, glucocorticoids and CRH suppress the release of GnRH or interfere with its pulsatile release, for instance, by acting on GABAergic neurons, kisspeptin neurons, and RFRP neurons. At the level of the gonads, glucocorticoids inhibit, generally, steroidogenesis. They reduce sensitivity of the gonads to gonadotrophins. And chronic glucocorticoids can induce apoptosis of Leydig cells, which are the cells in the testes that produce testosterone. So we have an inhibitory effects on multiple levels, both chronic and acute. The HPA axis response to stress is necessary for organisms to survive. However, repeated or chronic activation, according to the allostatic load theory, can lead to long-term health problems. So the idea is that excessive activation of the physiological stress response systems leads to wear and tear on numerous other physiological systems, which ultimately leads to disease and dysregulation. So according to the allostatic load model, individuals are particularly susceptible to the negative effects of excessive stress if it occurs during child development, which is of course a critical period for brain development as well as sexual development. So the effects of early life stress compared to stress at other points in development are particularly pronounced. Early life adversity has an impact on various aspects of health and development across the lifespan. Early life adversity is known to increase the risk of various mental illnesses, including internalizing and externalizing problems. It's also associated with an increased risk of various physical illnesses, such as cardiovascular disease and obesity. It is also associated with accelerated aging. We know from both animal and human studies that early life adversity alters cognition. It impairs learning and memory, affects reward processing, and can lead to impulsivity. Early life adversity also has important effects on social and emotional functioning. It's associated with aggression, with insecure attachment, deficits in empathy, as well as poor emotional control. Early life adversity has significant effects on sexuality and reproduction. It affects pubertal timing. It affects sexual behavior. While these effects generally vary between circumstances and individuals, generally, especially in humans, early life adversity seems to accelerate and disinhibit sexuality and reproduction. And there are a number of papers to support this. A recent meta-analysis was published by Ding and colleagues, if you're interested. Early life adversity affects puberty. In some cases, early life adversity may delay puberty. So this is often seen in cases of severe malnutrition and is often found in animal studies, particularly rodent models. Generally, especially in humans, early life adversity seems to accelerate puberty. This is supported by numerous meta-analytic reviews, studies done in large populations. It's also been found in a few rodent studies, but it seems to be specific to females. Early puberty in early life adversity survivors is important because early puberty is associated with an increased risk of certain cancers, such as ovarian cancer and breast cancer, testicular cancer, an increased risk of various mental health problems, accelerated cellular aging, and it is also associated with risky sexual behavior. In addition to affecting pubertal timing, early life adversity also affects sexual behavior. So in some cases, early life adversity, particularly childhood sexual abuse, can have an inhibitory effect on sexual behavior. However, in humans, more often we see that early life adversity seems to accelerate or disinhibit sexual behavior. Individuals with early life adversity start having sex at a younger age. They're more likely to engage in risky sexual behavior, such as having sex while intoxicated, having sex with unknown people, or having sex without protection. Early life adversity survivors also report a higher number of sexual partners. So, early life adversity in multiple ways is associated with disinhibited and accelerated sexual behavior. It's also associated with hypersexuality and compulsive sexual behavior. It's also linked to exploitative or coercive sexual behavior. And early life adversity is an important factor in etiological theories of sexual offending. So in addition to the effects of early life adversity on sexual behavior, it also affects reproductive timing and parenting. Early life adversity survivors have an early age at first birth. They have a higher number of children, and they are also more likely to engage in harmful or neglectful parenting. This has actually been found in both animals and humans. But wait, if early life adversity survivors exhibit accelerated sexual development and disinhibited sexual behavior, how can this be if stress has an inhibitory effect on reproductive function? To answer this, we must consider how early life adversity affects sexual behavior. So what does it do to the HPA axis and the HPG axis? But we also need to consider why, from an evolutionary perspective, does early life adversity affect sexual behavior? From the perspective of the allostatic load model, humans did not evolve to live in stressful environments, particularly early in life. Therefore, early life adversity is seen to disrupt development and lead to dysregulation of the HPA and HPG axis. Thus, the effects of early life adversity on sexual function, from this perspective, are seen as purely pathological. An alternative and more recent perspective is the adaptive calibration model. According to this view, humans did in fact evolve to cope with adversity throughout their lifespan. In fact, early life adversity, rather than disrupting development, redirects development towards a phenotype that is optimal for harsh conditions. This theory also attempts to resolve heterogeneity in how individuals are affected by stress and how early life adversity affects physiology. This newer model also tries to provide a more biologically rigorous account for the effects of early life adversity, drawing heavily from life history theory, which is a mature branch of evolutionary biology. So life history theory is useful for understanding how phenotypes might vary between individuals according to the level of harshness in their environment. Life history theory is based on the principle that organisms have finite time and energy to devote to fitness-related tasks such as growth, mating, and parenting. Because we have finite resources to invest in these tasks, this creates conflicts or trade-offs between different fitness-related tasks. The first trade-off is between long-term growth and survival and reproduction. So organisms must choose which one of these they prioritize. Because the evolutionary point of long-term growth and survival is to invest more resources that could then be spent on future offspring, we can rephrase this conflict as a conflict between future reproduction and current reproduction. The next conflict, nested within reproduction, is to prioritize either parenting or mating. Another conflict is between offspring quality and offspring quantity. So you can choose to produce a large number of offspring or choose to produce a few highly robust and competitive offspring. Strategic resolutions to these conflicts make up an organism's life history strategy, which varies between species and between individuals of the same species on a single continuum ranging from fast to slow. Organisms with a fast life history strategy have a greater number of offspring. They have a shorter lifespan, they produce at a younger age, and they tend to produce more offspring while investing less in parenting. Organisms at the slow end do the opposite. Critically, fast and slow life history strategy are adaptive in different environments. Fast is adaptive in harsh conditions where there is high premature mortality. This is because it increases the likelihood that the organism will leave at least one descendant behind, even if it dies prematurely. In contrast, a slow life history strategy is adaptive in a safe, stable environment where there is low premature mortality, because it increases the chance that the offspring will be more competitive and produce offspring of their own. So fast and slow are associated with distinct advantages and are specialized for different environments. Life history strategy is partly determined by genetics. However, it is probably plastic. This is because levels of premature mortality fluctuate far too quickly for organisms to adapt via genetics alone. Because of this, humans and potentially other species likely evolved to detect cues in their environment that predicts the level of premature mortality and to adjust their life history strategy according to this information. One cue that predicts premature mortality, or in other words, reliably covaried with premature mortality over the course of evolution, is early life adversity. So according to the adaptive calibration model, the physiological stress response systems, which include the HPA axis, play a critical role in encoding information that is relevant to life history strategies, such as harshness and premature mortality, taking this information and adjusting the organism's physiology in such a way as to change life history strategy so that it is optimal for the current conditions. So we know that the physiological stress response systems act on the HPG axis and other systems to change growth, maturation, reproduction, pair bonding, etc. So, high early life adversity is a signal of high premature mortality. So in response, this changes the physiological stress response systems or changes how the stress response systems communicate with other systems in such a way that it shifts life history strategy towards the fast end. This leads to changes in reproduction and sexual behavior that optimize fitness for harsh conditions. In contrast, low early life adversity is associated with low premature mortality. So organisms living in safe, supportive rearing conditions develop a slower life history strategy, which is associated with patterns of reproductive and sexual behavior and function that are optimal in safe conditions with low premature mortality. An organism's life history strategy has important implications for how the stress systems might affect the reproductive systems. A wonderful paper about this was by Wingfield and Sapolsky that was published in the Journal of Neuroendocrinology. So according to this paper, life history strategy dictates how stress, or if stress, affects reproduction. So remember how I told you that it makes evolutionary sense to inhibit reproduction during stress? Well, it turns out that makes sense only for some individuals. Specifically, it makes sense when you have high future reproductive potential, or in other words, you have a slow life history strategy. So if that's the case, it makes sense to postpone reproduction during stress and to wait for more auspicious, more favorable conditions. Thus, in this situation, the HPA axis activates in response to stress, thus inhibiting the HPG axis. This strategy does not make sense if you have low future reproductive potential, if you are living in an environment that is constantly harsh, if you have a fast life history strategy. If that's the case, don't postpone reproduction because you might not get another chance. So it's important in this situation to maintain reproductive capacity despite being in a harsh and stressful environment. In this case, it is therefore important that the HPA axis not inhibit the HPG axis. According to Wingfield and Sapolsky, there are a variety of tactics that organisms with a fast life history strategy might use to prevent inhibiting reproduction during stress. These tactics include downregulating activity of the HPA axis, thus disinhibiting the reproductive axis. Another strategy is to have compensatory activation of the HPG axis that overshadows inhibition by the HPA axis. A peripheral mechanism is to increase production of binding globulins that would bind to glucocorticoids and thus basically inactivate them. However, increasing binding globulins would not prevent inhibition of the reproductive axis via CRH, and it would not prevent glucocorticoids from binding membrane receptors, only nuclear receptors. So, Wingfield and Sapolsky give examples of specific species which show these different tactics. So the question is, do these tactics vary between individuals of the same species? And can particular environmental conditions such as early life adversity trigger these tactics? This is what I'm going to talk to you about today. For the sake of time, although there are many possible tactics, I'll be talking about two tactics that fast life history strategy organisms could use to maintain reproductive capacity under stress. Tactic 1 is to reduce HPA axis activity. Tactic 2 is to desensitize the HPG axis to the HPA axis hormones. These tactics likely vary between individuals and circumstances because they're associated with distinct advantages and disadvantages. So relative to tactic 1, the second tactic has the advantage of allowing the organism to mount an HPA axis response to stress. The disadvantage, however, is if you have joint activation of both the HPA axis and the HPG axis. This could be particularly bad for long-term growth and survival, according to Wingfield and Sapolsky, because joint activation of these axes might increase allostatic load and could, for example, lead to hypersuppression of the immune system. So because the costs and benefits might outweigh each other differently in different environments, we might expect different individuals to show different tactics. Let's talk about the first tactic, which is to reduce HPA axis activity, thus disinhibiting the HPG axis. We know that across vertebrate species, generally, fast life history strategy co-varies with lower activity of the HPA axis. However, findings are still inconsistent. So you still have examples of organisms with a fast life history strategy that do not show blunted HPA axis activity. This is also consistent with the observation that early life adversity survivors, in particularly humans, tend to exhibit blunted HPA axis activity both at baseline and in response to stress. But again, I must emphasize that findings are inconsistent, and many early life adversity survivors do not show diminished HPA axis activity. It is also interesting to point out that in rodents, early life adversity doesn't necessarily blunt HPA axis activity. In fact, it sometimes does the opposite, where it increases HPA axis activity. So the fact that we have these inconsistent findings could potentially suggest that organisms exposed to early life adversity or that have a fast life history strategy might use different tactics. So, blunted HPA axis activity is associated with various changes or characteristics in sexual behavior that are consistent with a fast life history strategy. So women who have a lower cortisol stress response reported an earlier sexual debut. In men, lower basal cortisol levels were associated with increased hypersexuality symptoms. Lower cortisol has also been associated with impulsivity and risk-taking, which could also contribute to hypersexuality or generally disinhibited or unrestricted mating strategy. This has important implications for how we look at blunted HPA axis activity in early life adversity survivors. So rather than seeing this blunted activity as evidence of dysregulation and pathology, it could in fact reflect a conditional adaptive response that helps the organism optimize their reproductive fitness in harsh conditions. There are many potential mechanisms for blunting HPA axis activity. We know that chronic stress or repeated HPA axis activation can epigenetically modify the expression of hormones within the expression of hormone receptors within the HPA axis. Specifically, we have epigenetic modifications of CRH and glucocorticoid receptors. So in mice, when we look at their pituitary, mice that have blunted HPA axis activity, we see lower expression of CRH receptors, which means that there's less feed-forward activation. And we see higher expression of glucocorticoid receptors, which means that there is more feedback inhibition. So these would both produce blunted cortisol responses. This is consistent with results done in humans, specifically studies using CRH administration or a dexamethasone suppression test, which also confirm that early life adversity survivors have increased feedback inhibition and reduced sensitivity to CRH. Another potential mechanism for downregulating the HPA axis is to upregulate activity of the HPG axis. This is because, as Victor Yao has reviewed, the HPG axis inhibits the HPA axis. So we know that sex steroids inhibit both CRH and ACTH production. It is also important to note that the afferent networks controlling the HPA axis, such as hippocampus, are rich in sex steroid receptors. So, chronic exposure to mortality cues, such as early life adversity, could lead organisms to change their HPA axis activity and consequently shift to a faster life history strategy. But this process of adoption could occur on a much shorter time scale. And we know this because when we look at birth rate following natural disasters and war, we often see this dramatic increase in birth rate that happens very rapidly, which suggests that humans may rapidly adjust their life history strategies when there's a rapid change in mortality levels. It is also the case that when you take humans in the lab and expose them to mortality cues, you see acute, probably temporary changes in their reproductive preferences that are consistent with a fast life history strategy. So for instance, after exposure to mortality cues, people show lower mate standards, prefer a higher number of children, and say they would like to get married and have birth at a younger age. My colleagues and I did a recent study where we took that same mortality cue and we exposed adults to this mortality cue before measuring their cortisol stress response. So we recruited men that either had low early life adversity or high early life adversity. So the people with low early life adversity, they showed blunted cortisol stress response. Among individuals with high early life adversity or low early life adversity, if they were exposed to the mortality cue, they showed a blunted HPA axis activity, suggesting that acute mortality cues could lead to acute changes in the HPA axis, which are consistent with fast life history strategy. So I would imagine that if these acute changes become chronic, so if an individual is chronically exposed to mortality cues, then these changes in life history strategy become entrenched. So life history strategy and the underlying physiological mechanisms could adapt to mortality cues both acutely and chronically. So are there tactics other than blunting the HPA axis activity? The possibility of additional tactics within the same species is consistent with the fact that early life adversity and fast life history strategy are not always associated with blunted HPA axis activity. A really good source of evidence comes from research examining coupling between the HPA axis and the HPG axis, which doesn't always exhibit an inverse association. What do I mean by coupling? So coupling refers to the within-person covariance of hormone levels from the two axes over time. So for example, I would measure someone's cortisol and testosterone levels in saliva repeatedly over time and see how they covary. So negative or inverse coupling is when the HPA and HPG axis activity change in opposite directions. So one axis increases in activity, the other decreases in activity. And this is consistent with inhibitory effects of the HPA on the HPG. However, this is not always observed. Sometimes we in fact see evidence of positive coupling, where both axes change in the same direction, so they can both activate or deactivate. We also see evidence of decoupling, where there is no longer a correlation between the activity of the two axes. Interestingly, we've found that early life adversity survivors, in fact, show evidence of positive coupling or decoupling, which could potentially suggest other tactics, such as compensatory activation of the HPG axis. It could also suggest altered crosstalk between the HPA and HPG axis. So the HPG axis could become insensitive to the HPA axis. This brings us to our next tactic, number 2, to desensitize the HPG axis to the HPA axis. So early life adversity could epigenetically modify the sensitivity of the HPG axis to the HPA axis hormones. A wonderful and very recent demonstration of this is by Granata et al., who took mice and used maternal separation to create a rodent model of early life adversity. So mice that had maternal separation showed downregulation of CRH receptors in both kisspeptin neurons. So this would mean that CRH no longer has an inhibitory effect on the HPG axis via kisspeptin neurons. So of course, consequently, in mice with maternal separation, we saw elevated levels of kisspeptin and elevated levels of GnRH. This accompanied earlier puberty, but interestingly, this was only observed in female mice. So it brings a possibility that perhaps there are sex differences in which tactics organisms use. This could potentially explain some studies in humans. So Meston and Lorenz recruited women that either had a history of abuse or no history of abuse, and they measured their cortisol response to sexual stimuli. Interestingly, among women without a history of abuse, higher levels of cortisol were associated with poorer sexual function. But in those women that did have a history of sexual abuse during childhood, there was actually no association between cortisol and sexual function. So this could, maybe, potentially, be explained by a loss of inhibition via the HPA axis on the HPG axis. It could suggest desensitization. I want to emphasize that I provided you with only a very brief, simple, and incomplete account for how early life adversity affects sexual behavior. It's important to acknowledge that the HPG and HP axis interact with each other in complex ways. And this interaction is shaped by a variety of genetic and environmental factors. It's also important to note that there are systems other than the HP axis and the HPG axis that are involved in altered sexual behavior among early life adversity survivors. We must also acknowledge that, of course, life history theory does not provide a complete explanation for the effects of early life adversity on sexual behavior. It is actually just one useful tool that you should include in your toolbox when we think about how early life adversity affects sexual behavior. So what's important about this life history theory perspective is to acknowledge that the effects of early life adversity on physiology and behavior are not necessarily pathological, but could reflect a conditional adaptive response which enhances their fitness in harsh conditions. So specifically, it could reflect a shift towards a faster life history strategy. What is also important to acknowledge is that fast life history strategy could have multiple physiological realizations. So this could help us understand why the physiological effects of early life adversity vary between individuals, because different individuals could show different tactics For some individuals, they might exhibit blunted HPA axis activity, whereas others could exhibit a desensitization of the HPA or HPG axis to the HPA axis. More research, however, is needed to better understand exactly why these tactics might differ between individuals. Could it depend on the level of adversity? Could it depend on sex? These are excellent questions for future research. Thank you very much for your attention.